overview of the HOAHub. And my first message is that it's going to be recorded, and so it is being recorded. If you don't want to be recorded in the video, please turn off your video. And we've muted everyone and asked that you put questions in the chat as we go along, and we'll answer them at the very end, as you'll see from the agenda that's showing. I'm going to hand over to Max now, who's going to start us off.
Yeah, good afternoon, everyone, and welcome to our annual general meeting of the HOAHub. My name is Maximilian Ackermann. I'm proud to be the co-chair together with Professor Peter Lee on this fabulous project, which we want to intend you to show in the next 1 hour. So our agenda for this meeting is, and you have just seen it, to give first you an introduction to the topic, what we're doing with the HOAHub, what our plans for the future, and what are the future options where you as users, collaborators, or interested people could contribute your ideas to our hub. Then we go over to some example, and then we come to the question and answer sections where you can really direct your points to us and ask what we can do. Please, the next slide. Yeah, what we're doing with the HOAHub. You see on this nicely picture, which was done by our colleagues from Siemens Healthineers, that we are planning first to do a visualization of organs on the one hand, and on the other hand, we want to implement the HOAHub as one platform for to do a correlation with other techniques. Next one. What is our vision of HOAHub? The HOAHub started with the pandemic of COVID-19, and at that time, a different team formed out of interdisciplinary people, physicists, anatomists, pathologists, which came to one table to make this vision real. On the one hand, to do from autopsy tissue fabulous pictures and movies, and on the other hand, to implement a HOAHub as one technique which could resolve also clinical questions. So this is the next step for us for the HOAHub, to embed HOAHub as a clinical routine platform where you can also do a correlation or correlative imaging with other established methods. And the aim of the team, the aim of all our project is to do an open access platform where we can contribute shared results, methods, hardware, and software. So it's a real hub where you can learn from each other and to share your vision and to share your ideas with your colleagues. Please, the next one. Yeah, the HOAHub was created as a bridging technology because as an anatomist and pathologist, we are just limited to light microscopy. And on the other hand, our colleagues at the Department of Radiology are really looking with a low resolution on the organs, but they cannot contribute really the morphological changes which could be detected by us by the microscope. So the HOAHub is really the bridge between those two disciplines, from radiology down to pathology or histology. So you can see here on this nicely movie the reconstruction of blood vessels of a lung of COVID-19, which was the starting point of the HOAHub. You can nicely see the cross circulation between the different veins and arteries. And so it could help really to do a multi-scalar view on the organs, but also a multi-dimensional view on the organs. And the nice thing is that if you have gathered all the data and you have a platform of all the data, you can also learn by AI from the different morphological changes which are detected in the different methods. Please, the next one. And this is nicely seen here on the reconstruction of a brain. So you see on the left side the classical 3 Tesla MRI scan, and then the development of the HOAHub, which started with on the BM05 down to BM18, where the resolution is really fantastic. And you see here the brain stem, and with the segmentation of relevant structures of the brain parenchyma, you can also then detect changes in the vascularity, changes in the gray matter and the white matter, and thereby do a correlation then also with the existing clinical MRI scans. Please, the next one. And then I would like to hand it over to Professor Lee.
Great, thanks very much, Max, for that introduction. Reminded us the amazing things that HIP-CT can do. So really, the specific examples of goals of HOAHub are really to enable the accelerated development of HIP-CT, hierarchical phase contrast tomography, and make sure that it's applied to the world's really most needing biomedical challenges. And we had some specific goals, and the specific goals were very, very much to first solve key biomedical challenges, to help better understand and mitigate and hopefully cure some diseases. We also want to do multimodal imaging, correlating upscale to, as Max mentioned, CT, MRI, and other imaging techniques, and go downscale. And we'll show some examples later on, all the way down to omics. So connecting through from whole organ down to molecular. We also want to try and translate some of that to clinical applications. It would be brilliant to help get really higher resolution or super resolution in MRI and CT using HIP-CT. And we're looking at working with laboratory sources, people like Sandra Olival, on how to translate it into the lab so it can be used for 3D histology in hospitals themselves. And finally, we're hoping to enable groups worldwide to use HIP-CT. In fact, we've had over 70 scientists come and train on HIP-CT since we started 3 years ago. And finally, David, at the end of this talk, is going to show you how to use the open access data. So all of the data on first publication or 3 years, whichever happens first, and hopefully it's first publication, is put into the open access. And also anyone can ask for any of the non-open access data. And so long as it's not being used in a directly conflicting manner, we share it out. So really that dissemination, there's 10 co-PIs. So there's 10 of us who all equally are running the hub. Each of us with different skills and different interests. And really that's what the ESRF hubs are about. They're about bringing together a whole set of interdisciplinary, complementary skill sets to challenge some of society's greatest needs, such as understanding disease and how we age. And that's really the vision of the HOAHub. It's in a nutshell to help mitigate and cure neurodegenerative diseases to cancer, so all types of disease. And we found now that we've applied it to over 30 different organs and many, many different diseases, dozens and dozens. We're also hoping to use it for training, public engagement, and going through and propagating it, not just from ESRF, but to other facilities as well. And ESRF has said that they're very happy to help train any other facilities. So we've had APS on site and from groups from Korea and many other places to look at how HIP-CT works. What has it actually done? Well, to date, we've scanned over 1,000 data sets, half a petabyte of data, 300 organs from 200 donors, with over 30 organ types. As an example, and these numbers are already outdated from our last beam time. We have 57 hearts as an example, 39 kidneys, 4 colons, 4 prostates, many more than 4 prostates, placenta and other organs is listed here. All of that now from the HOAHub, which began in September 2023. So really just a little bit over 2 years ago from the formation of the proper hub. We've helped over 70 scientists, as I mentioned, come to Beamlines and be trained, going through and that data and knowledge going to over 70 groups worldwide who are either collaborating directly or using the data. And for everyone, how to use it and what are our outputs? Well, to date, we've had over 30 articles, which are all listed at that URL listed there, mecheng.ucl.ac.uk/hipct/publications. And we've had over 10 articles published from groups who've downloaded the data, written up a publication on it, and got it out. And you'll find all of that data at human-organ-atlas.esrf.eu, as David will show examples later. That is a public database. As you'll see is this example snapshot from that database, a number of organs. We're continuously putting up new data going through. And if you don't see something that you might want, please just ask. So how do people who aren't actually one of the 10 co-PIs use it? Well, for anyone, you can use that data. And we've got David who's going to spend 15 minutes to show you how to do it. You can get trainings. We're happy to train people. The easiest way is to come along in a beam time and see how it works. If you've got something where we've got a biomedical challenge and we haven't scanned that type of organ yet, then we have something called feasibility beam times. So anyone can apply. We go through and run them for you up to 1 shift or 8 hours of scanning, which will either do a number of organs in an overview, sort of a 20 micron, 15 to 20 micron resolution scale, or do maybe 2 organs with quite a number of zooms down to 4 or 2 microns. And we do that through proposals. To get information on those proposals, go to that website, or when you clicked on the Eventbrite today and said you're happy to get further information, we'll add you to the mailing list, which you can remove yourself from, but you will then be emailed with every call for proposals. Finally, for the 10 PIs and core collaborators, we have something called biomedical challenges where you can get normally 1 to 6 shifts, can be more. It does have to have one of the PIs on it. The reasoning behind that is that it's not a simple technique. And so in order to make sure that everything works, you need to collaborate with one of the 10 groups who are using it regularly. Any questions, please put them in the chat. And I'm now going to hand over to Joseph, who will tell you a little bit about some of the achievements technically that we've made. We'll then have Bernadette, who will tell you about some of the uses that she's made of it over the last year. Joseph.
Yes, I'm sharing my screen. It's there. Do you see the screen? Oh,
Yes.
maybe I should do that. Perfect. Hi, everyone. So, Joseph Brunet. I'm a postdoc in Peter Lee's team, and I'm coordinating the activities of the HOAHub at ESRF. So I will present you quickly the achievement the technical achievement that in the past year. So I chose a few ones. There's more, but I chose the one that seems to me like the most important one. First, I'll talk a bit about sample preparation. So sample preparation got quite improved thanks to Joanna, our lab engineer, in the past few years. We used to do formalin bath, ethanol bath, agar mounting, and degassing. Now, thanks to Joanna that streamlined everything, we mostly do in formalin, so we gain 3 weeks in the whole pipeline. And she worked a lot in the agar mounting and the inline degassing. And thanks to her work, now we are able to mount an organ and to degas it in a day, and sometimes a bit more, but around a day. And that's unlock a lot of possibilities like scanning organ in clinical pipeline. So for example, after a surgery, the explants can be sent to ESRF, prepared, scanned in less than a day or two, and sent back to the hospital to be processed by the normal clinical pathway. So it unlocked quite a lot of possibilities. And this is mostly due on the work on the degassing side that took a lot of time before. So if you don't degas well the organ, you obtain something on the left where you have a lot of bubbles. And it used to be a huge problem that we have where most of our scans were full of bubbles. Not most, but I would say quite a lot. And the technique that we used to degas was this one, where we just remove the bubble by vacuum degassing. And thanks to Joanna and the work, now we have this kind of setup where it's inline degassing, where we have 4 pumps and a membrane degasser, where we can degas the organ inline and parallelize it. So like we can degas 4 organs at the same time. And we have a few of these setups, which allows us to degas a lot of organs before beam time and be prepared to avoid bubbles, to reduce problems. So we increase our capacity in scanning quite a lot because of this. Now, if we look at the scanning aspect, there's a lot of new things on the beamline. I will focus on one that changed our life quite a lot, which is the detector stage. So we have now quite a lot of new detectors. And one of them is the LAFIP, which we had for a few years now, but we really use it to its maximum capacity now. It allows us to scan a complete organ at 16 microns in 1.5 hours. So here we scan 8 hearts in less than 12 hours. So it increased quite a lot our capacity. We can scan lungs, heart, brains in 1.5 hours. You can see the reconstruction time also is around 2 hours. I will speak a bit about it later. We worked quite a lot, and it gives you volumes between 1 and 2 terabytes, and the contrast to noise ratio is still quite high. So we keep the quality that we had before, but we scan them much rapidly. To go quickly, the improvement, there's many, but the main ones will be helical scanning that we use most of the time now. There's larger cameras, and we received recently even new camera, the PCOH10, where we will increase our speed by a factor of 3, so it will be even faster. And we have more efficient optics and scintillators, which now the main limits in scanning speed is the transfer speed of the camera, which we will unlock with the new cameras that we received. In terms of the pipeline to scan things, there was quite a lot of work at ESRF to on our side to automate this scanning, to automate the scanning by creating automated scanning macros. So you can see an example on the right. You will fill the parameters of your sample and that you would like the jar size, the patient name, organ name. You put your sample on the sample stage, you launch the macro, and it will guide you through the different steps like alignment, filter optimization, etc. And it allowed us to reduce the errors drastically in terms of naming the scan, in terms of alignment. We can also launch the scans in minutes. Before it was 10 of minutes or hours. And it's also easy to teach users and people that come on the beam time to launch scans. So if you came on the beam time, you probably use one of these, and it's really easy to use. In terms of reconstruction, it's a bit the same. We developed a command line interface pipeline to streamline our reconstruction and make it much easier and parallelizable and faster. The pipeline called Nightbus. Compared to before, it's more reliable, it's faster. As you saw, we can reconstruct whole organs in 2 hours. Before it was, I think, between 10 and 20 hours when we use MATLAB. And it's easy to teach users. We start to have users from UCL that know how to run this and they can reconstruct their own scans with minimal help. And it has been adopted by other teams at ESRF to reconstruct, like teams in paleontology or in material science. In terms of hard tissue, so we showed a lot of organs and soft tissues, but we focus a lot on hard tissue also. So this work comes from a paper that was released a few years ago, but published this year by Alessandro Mirone on econol phase retrieval, where here you see a sheep head reconstructed with the classical Paganin algorithm. And here with the algorithm that we developed, so the econol phase retrieval, to cope with the artifacts created by the hard tissues. So you will see that in the brain case, you have much less artifact on the right than on the left. And that allows you to do better analysis and more precise analysis. This enable a work on the sheep head done by Teresa Urban that will be submitted soon. And you can see the beautiful images that she was able to create thanks to this. Another things that is quite a big achievement is the use of the large sample stage. So it's at BM18, we are limited to a small sample stage that allows us to do maximum 50 centimeters high, the sample. Now we can use a large sample stage, and we were able to do a whole spine that was 80 centimeters high. And you can see here the results. We scanned it at 16 microns and did zoom at 2 microns locally in areas of interest. And you can see that we see very well the hard tissues and the soft tissue thanks to HIP-CT. And this is just the beginning. The sample stage had some problems in the past few months, but it should be solved now. And we will use more and more either to scan large objects, very large objects like spine, like whole body, like other things. But we'll use it also to stack organs and to streamline our scanning, to scan always faster and go toward large N numbers. And here you can see a part of the work of Ayla Anderson, where she started to segment some vasculature in the spine and some nerves. So it will be quite interesting. We also have something that is quite nice and more teams are using is multimodal application, because HIPCT gives you a lot of nice morphological input on your samples, but you could have other input with other techniques. For example, a lot of teams are using combining HIPCT with transcriptomics to have cellular information. So this is part of the work we did with OCMI, where we scan the heart with HIPCT with these chunks, and then we scan these chunks again, and we send them to Cambridge to do histology, and then to do xenium staining, to finally obtain the cellular information in the chunk. So we can really correlate HIPCT with the cellular activity at an instant here, which opened quite a lot of new insights and possibilities. Oops. And finally, for the future, so as I said, we are going through large N number. We want to have a very biomedical impact, and for that you need statistical significance. So we'll always strive toward large N numbers. We started a few studies where we scan, for example, in tetralogy of Fallot, we scan between 15 and 20 hearts with this disease. But we continue to go in this direction to target some disease and having larger N numbers. We'll use more the large sample stage, which will allows us to scan larger things and also go faster for this a larger number. And finally, we'll go toward anatomical pieces. We scan a lot of organs. We started the anatomical pieces with the spine, but we hope to continue to scan even more and added them to the HOAHub database. And I think I finish here, and I will hand over to Bernadette.
Great. Thank you very much, Joseph. And just a reminder to anyone, any questions whatsoever, please put them in the chat and we'll answer it at the end of the talk. It's my great honor to introduce Bernadette de Bakker, who will start showing you some of the amazing applications that they're doing at Amsterdam Medical Hospital.
Great. Thank you, Peter, for the introduction. Can you all see my screen? I guess so.
Yes, we can.
I'm one of the main PIs of the HOAHub, and today I would like to tell you about the improvements we made within the Amsterdam UMC team to unlock unique opportunities for biomedical research. Let's see. So this is my team in Amsterdam. I have several PhD students and brilliant technicians working mainly on human development, embryology, because I'm an embryologist, medical doctor with an embryology background. And I've also worked for 16 years at an anatomy department. And because we set up the Dutch Fetal Biobank a couple years ago, we have a lot of experience with collecting fresh samples from different hospitals in the Netherlands, fresh embryonic and fetal samples, and how to store them the best we can to allow as much as imaging and microscopic analysis possible. And that's why I think I was involved 2 years ago within the HOAHub team. And because it is the second hub evolved at ESRF. And it's really brilliant to see how all these collaborators have this great goal in mind to share knowledge, technology, and beam time. And these results are really happily shared within the team. I will tell you today about several organs we have scanned within the Amsterdam team. You see them here on the screen, and I would like to tell you more about the challenges we've overcome. So the first challenge was the post-mortem interval of the tissues that we've been using within the HOAHub. Most tissues come from LADAF, the French anatomy department. Often these bodies are dead for a couple days before the organs can be harvested. And to allow single cell resolution imaging, we also would like to have the best quality organs possible. And in the Netherlands, the laws are in such a way that we can ask people who would like to donate their body to science. Well, if they want to let us know up front if they go through euthanasia. So then we get a phone call up front that we can expect a body. And that's why and how we collect bodies which shows short post-mortem interval. These include embryos and fetuses, but the adult tissues are scanned within the HOAHub. And this is done mainly by my colleagues, Jermo and Daniel. So we get a phone call, usually during the week, that we can expect a body, and it can be in the morning, afternoon, evening, or night. Our team is 24/7 on call to start dissecting these bodies immediately when we get them, to make sure that even when the body is still warm and the tissues are very fresh, we can store them on paraformaldehyde. And this also allows every histological or immunological or spatial transcriptomic analysis after imaging. The second resource we have are surgical samples from our institute, for example, of the colon and the uterus. I will show you later. These are all patient samples, and these patients approve for biobanking of the tissue that has been removed during surgery. The second challenge we had to overcome is the ethical challenge. Most of us really have to deal with that, but luckily within the HOAHub there's a lot of help from Rukang, for example, who really helped us through the French transport ethics and the ESRF ethics. So from our institute we are able to send whatever organs towards ESRF for being scanned. This point, the mounting and degassing has been shown at ESRF by Joseph, who just presented it. And we also chose to build our own mounting and degassing lab in Amsterdam, also to help out the team at ESRF to avoid that they have to mount and degas all organs of everyone. So we're happily mounting and degassing ourselves, and these spots are then packed and sent towards ESRF, where they are put in the inline degasser just for another couple of hours before they can be scanned. So there's no handling of the tissues anymore by the ESRF team, which is very helpful. So then we go scanning, happily scanning. We've had several brilliant scanning sessions already, but then you have huge amounts of data. And what to do with the data, of course. The first one is that we prefer to do correlative imaging of the data, not only with patient scanners, but also with, for example, micro-CT. And I just want to give a brief example, which is the colon. This is the PhD research by Daniel Doctor. And he managed to put a whole human colon in this jar. Not in the anatomical position, of course, but still it could perfectly be scanned. And if we zoom in a bit on specific region of the colon, you can see really that we see single villi of the gut. And within the villi, we can see the individual cells who are producing the saliva of the colon. The next one is an even more higher resolution image. And what we see on this zoom scan of 1.4 micrometer is apart from the different layers of the gut, the submucosa, the circular muscle, for example, also the nerve. This is one of the nerves of the enteric nervous system. And this data really helps us to understand better how a human colon looks like. And of course, we cannot send all of our patient samples to ESRF. That's not feasible because it's much too far away, and we don't have that many beam time shifts. But luckily in Amsterdam, we were able to install our own micro-CT system here in our radiology department. And this really allows us to scan also patient samples. So this is an example of colon scanned by micro-CT. And again, if we compare the micro-CT imaging with the histology, it really helps us to see all the details we need in those patient scans. And this, correlative imaging between HIP-CT and micro-CT really helped us to understand, for example, how the myenteric plexus, so the nerves of the colon of patients with functional constipation, looks like. And it appears that they look different from the nerves in healthy patients, whereas doctors in the past always thought that functional constipation is between the ears of the patient. So this was really one of the examples where we made a lot of progress in correlative imaging. The last example I want to show is how we use AI solutions to tackle the big data segmentation. That's done, for example, in the uterus, but also in the colon, as I showed you before. This is the work of Veerle, one of the PhD students, and she's studying the uterus, and especially healthy uteruses she compares with patients with adenomyosis, which causes more bleeding and more pain during menstruation. We have scanned these whole organs at 20 micron resolution with detailed zooms at 2 micron. And we have a whole team of students ready to do the manual segmentation of all the structures and blood vessels, nerves within these organs. And I specifically mention this because we have many brilliant postdocs who are really eager to help us medical doctors to see or to detect the structures by using AI. But someone has to train the AI, and that's really what we invested in the last year, also together with the group of Joe Jacob, the group of Peter Lee in UCL. They really helped us with using the AI, and by going back and forth between the two teams, we were able to train this AI to show in a human uterus all the blood vessels, both the arteries and the veins that we have been looking for. And it's way too much work to do that all manually. So I'm very happy that we have such a great AI team within the Human Organ Atlas Hub. That brings me to my last slides, the take-home messages of my part. So we set up short PMI tissue biobanks to collect organs. We do correlative high-resolution X-ray imaging within our institute. And I must stress, this international network is really brilliant, and collaborating within the HOAHub is truly a great experience for my team. And thank you for that.
Thank you so much, Bernadette, for showing an amazing set of examples of how the data from HIP-CT, coupled with other techniques, can really go through and help us understand disease. I'm going to now hand over to David, who will go through and show you how you can download some of the data and use it yourself. Do remember that it's only about a quarter of the data that's currently up there. We're continuing to put it up. If there are things that you'd like to know, please email and we'll try and do it. Or ideally, put a question into the chat. David, hand over to you.
Thanks a lot, Peter. So, hi everyone, I'm David Stansby. I'm the data scientist for the Human Organ Atlas, and I'm going to give you a quick update on our public data, walk you through how to download it and start using it yourself. So here are some beautiful images from some of the many data sets that you can download and start using today. This is a graph of how much data we've generated over time in blue. So starting back in 2020, when the project started on the left, all the way through to now, almost 2026, we've generated over 600 terabytes of data, and we've released over 100 terabytes of that data. So as Peter said, that is the data is released either 3 years after it was taken, or hopefully sooner on publication. This is a similar graph showing the size of each of our data sets, again from 2020 on the left there up to the present day. And this is really to emphasize we're generating huge volumes of data. We're routinely generating images that are over 1 terabyte. And so there's a really exciting opportunity there for groups around the world to download this data and mine it for all the information that's in there. This shows an overview of all the data sets that we have publicly available. So in the middle there is a list of all the different organs. The ones underlined are new organs where the first data sets were released in 2025. So we had our first colons, prostates, spleens, testes, and uteruses released this year. We have lots of overviews for all of these organs and many Zoom data sets. So the overviews you'll typically find at 20 micron or 16 micron resolution, and the Zooms 4 micron, 2 micron, and in some cases we even go down to 0.8 micron voxel size here. So if you want to download any of this data and use it in your own research, this is our website, human-organ-atlas.esrf.fr. And now I'm going to take you through going through that website and finding some data that you might want to use. So this is the landing page. It gives a bit more information about the project. The easiest way to go looking for some data is clicking on Explore here at the top. And then we have 2 ways you can look at the data. If you're interested in researching a particular organ, you can use this organ tab here, and we list all the organs we have available. And if you click on one of these organs, you can then see all the donors where we have samples available for that specific organ. If instead you're interested in the same organ, different organs for the same donor, you can click on the donor tab here and see for each donor which organs we have available. So for example here for LADAF 2021-17, we have a whole load of different organs available to look at for that single donor. So I'm going to go back to the organ tab here. If I click on one of the organs, we can then see all the donors. If I then click on the donors, this will go through and show us all the data sets available for that organ and donor. So in this case, all the kidney data sets available for LADAF 22-27. If I click on one of these data sets, we then go to the data set page. So this gives you an overview of everything we know about this data set. So a brief description, a citation, a nice visualization there on the right, and then at the bottom here we have all the metadata. So you can see how we scanned this image, sample information, and other stuff that is important to contextualize the data set. The next step, once you've selected a data set you're interested in, is to visualizing it. And we have options to really easily visualize the data. So if you click on this NeuroGlancer visualization new tab link, a new page will open in the browser. So this doesn't require any software. This is entirely browser based. And already you can see an overview of that image showing up. So in the bottom left here, we have a 3D view. And if I turn on the sections, you can see the sections that are being displayed in the other quadrants. And so we can zoom out and look at this organ at the full scale. And then as we zoom in, the data will load at higher and higher resolution. So you can see now that that's loading over the internet. And so you don't need a high-spec computer already to really dive into the data, visualize it, and see if it's something you're interested in downloading for future analysis. So once you've done that, how do you download the data? On the right here, we have links for all the different ways to download the data. At the bottom here, you can download a metadata file in JSON format. And so that gives a machine-readable version of all the metadata you saw on the web page before. And then you can download the data at multiple resolutions. So at the top is always the full resolution data. And then we have downsampled versions if you want to download smaller data volumes at lower resolution. For the smaller data sizes, so under 2 gigabytes, you can just directly download a zip file of the image stack through your browser. And for larger data sets, so greater than about 2 gigabytes, we have a Globus link here you can use to download the data using the Globus application. And that's much more reliable than downloading it simply through your browser. And then finally, once you've downloaded the data, done some analysis, written a paper, we have here a really easy citation, so you can give credit to us for generating the data. And in return, I'll explain later, we're going to start listing publications that use our datasets on this website. So by giving credit to us, we can give credit back to your publications that are using the data. I'm going to just finish off now by talking about a couple of advanced ways to work with these huge data volumes. If you're interested in mining the metadata in a machine-readable format, we have all the metadata files available on a GitHub repository. And then we also have a set of tutorials. This is on the website on the Help tab. So if you want an extended tutorial on Euroglancer, how to use the data in Fiji or in Python, this is the place to go. And then finally, if you're a Python user, we have a Python package called the Human Organ Atlas Python tools, which you can use to interact with the volume data in a very advanced ways. So to finish, we have just a roadmap for the coming year. What features and data are we looking to add? We're looking at adding links to load the data directly into WebNosos, which is an online platform for visualizing, sharing, and annotating 3D images. We're going to start uploading derived data sets, so segmentations and meshes. So on the right here, you see some of the segmentations of some of the kidneys we've done. We want to add those to the portal. We're going to link to multimodal imaging of the same samples. So both Joseph and Bernadette talked about this, and we're going to start linking our datasets to other modalities. And then linking the datasets to related publications. So when you give credit to the dataset you've used, we also want to give credit to your paper that has used that dataset. So we're looking to list those publications on our website. In 2026, this is a plot of all the datasets we plan to release. So lots of brains, hearts, and lungs, but also of the soleus muscle and a few other organs. 2 datasets that are particularly worth highlighting are a heart that we scanned at 8 micron resolution and a brain that we scanned at 7 micron resolution. So these will be our biggest datasets yet, coming in at about 15 terabytes each. So I'm going to finish there. Thank you for listening, and I'll hand back to Peter to introduce Gaush.
Thanks so much, David, for giving us a great way of showing how you can use the data. And we're going to now go on through and take in a new member. So the HOAHub is open to new members. Gaush is a new PI who's joined just in the past year. The whole goal is to bring in people who are complementary and can really add. And that adding can be through new analysis, new sets of organs, new disease challenge, and hopefully provide funding to keep the whole thing running. I'm now going to hand over to Gaush.
Good afternoon, everyone. Thanks, Vita, for that introduction. So my name is Gowsihan Poologasundarampillai. I'm a reader in biomaterial science at King's College London. So it's a pleasure to speak briefly today about my personal journey with the HOAHub, specifically how I transitioned from being a curious external user to a core PI of this incredible community. Our research focuses on critical yet understudied areas of women's health, Specifically, we're working on a welcome leap in vitro program to prevent stillbirth, and also on a UKRI CRCRM program to model lung diseases that predominantly affect women of childbearing age. If we take the placenta, for example, it's a critical life support system for the developing fetus, yet it remains poorly understood. Globally, every 16 seconds, one baby is stillborn, and placental dysfunction is a major driver of this tragedy. The challenge is that the placenta is a large, soft organ with a hierarchically structured, complex, heterogeneous network of blood vessels and villi. To identify novel biomarkers and to reduce preventable stillbirths, we need to map this structure across scales. This is where HOAHub is indispensable. It complements our multimodal workflows by providing the only imaging modality capable of capturing this soft hierarchy in 3D without destruction. It bridges the gap between our clinical questions and clinical modalities and the successful translation of our approaches to clinical solutions and to patients. The fast track mechanism, which can be obtained through either training and feasibility study beam times, give us the perfect gateway to generating first data set on your clinical tissues of interest. Here, I'm showing our first beam time, which was a feasibility beam time performed in January 2024 on 3 small lobules of 3 different placental lobules. And one of the aims here was to look at the different sample preparation and how that affected tissue quality. And the first data set that we got through from HIP-CT proved that the level of resolution and the contrast achieved were sufficient and more than sufficient to resolve most of the features of interest. And here I'm showing a video of a transverse GIF file showing how from the chorionic plate, how the blood vessels branch down to the other side of the placenta. And what we're highlighting with the arrow is this central cavity, which prior to this have been shown using SCM corrosion cast and schematics, which were obtained in 1972. But with HIP-CT, with a single image of a lobule, we can start to see these complex structures quite clearly. So once we had this initial confidence, this collaboration evolved into a biomedical challenge to be able to image entire placentas. Our clinical collaborators, so Paul Brownville, who's probably not on this call, and others who are on this call, took on this challenge of preparing entire placenta. So we went from looking at small lobules, which are of the size of a burger, to preparing entire placentas, which are about 20 centimeters in diameter and 10 centimeters in height. Working with HOAHub, especially Peter Lee and other co-PIs and staff within HOAHub, we were able to image 3 whole placentas, 1 normal healthy donor and 2 gestational diabetic placentas. So this was through a biomedical challenge, which allowed us to image the entire placenta over a day of beam time. So just as Bernadette was describing, we were able to replicate some of the sample preparation, so degassing and placing into these jars, which were then safely transported to ESRF, where further inline degassing was done. Prior to that, we were able to generate some ex vivo MRI dataset of the same placenta, and at ESRF, we performed HIP-CT, generating overview images of the whole placenta and multiple zooms going all the way down to being able to resolve terminal villi. So we were able to go from looking at the whole placenta, small cotyledons, down to stem villi, and then intermediate villi and the terminal villi, all from a single modality. So this transition from a user to a collaborator naturally led me to becoming a member of the HOAHub. And I see myself as a bridge between researchers with really interesting clinical problems, researchers and clinicians with really interesting clinical problems, and imaging modalities. And here I'm showing a couple of projects that I am acting in this role. One, which I briefly spoke about in terms of imaging placenta and coming up with biomarkers to prevent stillbirth, and also the other project funded by UKRI, looking at imaging lung tissue to be able to model lung tissue destruction, and both of these projects rely on this hierarchical soft tissue imaging that HIP-CT provides. Having becoming a co-PI at HOAHub, I realized that to get the most out of this technology, I need to contribute to this ecosystem. And so therefore, I'm involving myself through presenting at several meetings at the HOAHub, reviewing proposals, and actively working with other members to try and develop new proposals to engage new users to this really vibrant community. So finally, I just want to end there to thank all the people who supported activities within HOAHub and my collaborators who have provided so many access to tissue and as well as access to data and processing. And I saw there was a question on placenta, which maybe we can talk about during the question time that's to follow. Over to you, Peter.
Thanks so much, guys. Sorry, I was unmuting myself. Going through. Thank you so much, guys, for an amazing talk and showing really how you can start engaging at a simple level, an entry level, and get more and more complex and gain more and more out of the HOAHub and the interaction with all of the different PIs, which Gauss is now working with around half of us. I think at that stage, it's now time for questions and answers. So I'm going to go through and start off. There's a question for you, David, if you want to start by answering that.
Yeah, so the question is, does the metadata schema you describe HIP-CT data only describe HIP-CT or the multimodal data as well? Our metadata schema is specific to HIP-CT and specific to how the data was acquired, so it doesn't describe the multimodal data. Something we'll be working on in the coming year is how we make sure our schema is interoperable with other schemas. So where we're, for example, describing donor metadata, making sure we're describing that in the same way that other schemas would do.
Thanks very much, David. If anyone has more questions, and I'll carry on with the ones that are in the chat, you can put up your hand as well and then unmute and go through and turn on your video if you want to ask the question direct. I'm going to quickly answer one question myself, which is on the presentations. They will be available. They'll go onto the HOAHub website, which is in the chat. The next question, I think Gauss has volunteered to answer, which was how, if you want to access data which isn't yet made public, how would you do it? Do you want to, which in particular is the 6 placentas that have been scanned. Gauss, you happy to answer that?
Yeah, I can take that, and maybe please add to that from a general HOAHub policies, Peter and others. With regards to placentas, we have scanned 6 placentas. 3 of them are smaller lobules, and 3 are the whole placentas. At the moment, our teams are going through and analyzing that data set. So first scans were produced in 2024, and the idea is to make those available as soon as possible, as soon as we've had a chance to go through all that data set ourselves internally for the funded projects. And I think there's a general HOAHub policy on when data becomes available to others.
I'm just going to add to that, Gowsh, that actually the policy goes further, that if you are interested and the data isn't published, you can email through. We'll then have the people who assemble a small group, which will be one person from the exec committee, one person who was part of the scanning and own the organs, and just get them together and make sure that if you have a use which doesn't actually contradict but complements the use of the HOAHub PI, will release the data. And so far, in all cases, we've released the data. So we've always managed to go through and look at some way to be complementary and actually make it benefit for both groups, those who did the scans, or multiple groups, those of us who generated the scans, those who provided the organs, because a lot of work goes into it, as well as those who analyze it. I hope that answers your question. If not, you're welcome to put up your hand and go through. Next question goes through to cranial nerves. I don't know whether Teresa wants to answer that one, but we are starting to look at scanning the human head. And I don't know, Teresa, whether you were able in the sheep's head to see cranial nerves. Oh.
Yes, we have seen nerves, but I have to say I'm not an expert in anatomy, so I wouldn't know if we have seen the nerves, especially at the level of detail.
I can help out there.
Interested in? Yes, please.
No worries.
So it sounds like something that if you're interested, drop an email. Rickon, if you could put the central email for the HOAHub there, which is hoahub@gmail.com. Go through, drop an email, and we can assemble a grouping and take a look through and at the images. And we are also going to make the sheep's head available as soon as we submit the paper, which should be very soon. So you can walk through the data yourself and have a look. Absolutely, in things such as the spine, you can see the nerves. They're not easy to trace. And I think those, Joseph's nodding his head, and he'll nod his head that they're not easy to trace. You can see them. Quantifying them is another challenge. And that's why I think Bernadette talked a lot about the AI, and lots of the people in the group are working on AI to capture these. If we have any other questions, please stand up. Yes, the data sets are also available for teaching purposes. I don't know whether, Max, you want to give an example of the use of data set that you're going to be doing very soon?
Yes, so what we are doing with the data sets, we'll be using that for our medical students to highlight their, on one hand, the anatomy, to show really movies of, to fly in and have a deep dive into the lung, heart, and other organs for anatomical purposes. And on the other hand, we also highlight this in correlation with our teaching on pathology to do a multimodal approach to histology and diseased organs. And on the other hand, yeah, we are planning actually also an exhibition on those purposes.
And I see Lisa's on. Her supervisor, and I'm not sure if Lisa wants to speak about it, but Lisa Chestnut, her supervisor, Andrew Cook, uses virtual reality headsets to go through and train to look at studying at the case of pediatric disease in the heart and how you could do surgery. Lisa, do you want to give some details, because you know better than me?
Yes, I don't know a massive amount, but there's VR used for teaching and communications, and we've done some conferences allowing people to come from lots of different centers to learn about congenital heart disease in that very immersive environment. They found it's very helpful for teaching.
Great, thank you very much, Lisa. I hope that has answered the question. Any other questions at this stage? Put up your hand. We have a couple of minutes left. We're running over, and then Max is going to close. If there's no other questions.
There's one question on the optical nerve.
Yes, so that's back to Teresa if you want to go through. We have some beautiful images of the optic nerve.
Yes, so we have in the sheep's head image the optic nerve close to the eye and close to like where it goes into the brain. We haven't done that in a human head yet, but generally, yes, it is possible.
And the results are really lovely. You do need to zoom down to 2 or 4 micron resolution. So you have to, it does take time because you have to scan along the optic nerves. And they are quite gorgeous, even though I'm not an anatomist. I think with that, we'll go through and close overall by handing over to Max to do a closing summary.
Yeah, thank you very much, Peter, and thank you also for the questions you put on the chat, and also that was raised by your personal questions. So to conclude, I would thank you all the speakers for this wonderful afternoon and to the overview you have given to us, and also the chance and opportunities that are made possible by the HOAHub. So for us, it's important that we stay in touch, that you ask us your questions, and that we can help you to support for the realization of your projects and your biomedical questions. And therefore, yeah, please ask us more questions. Please submit your applications for the next Beam Time. And we are really, as I said, happy to support you anytime. Thank you so much and have a great day.
Thanks, everyone. And again, the email is hoahubesrf@gmail.com. Thank you, everyone. Bye, everyone.